screen visible to all of you? Aisha, Parker, Rabia. Okay. So showing me my connection is unstable. I hope my voice is clear, right?
Yes, sir.
Okay, please let me know if it breaks down at any moment. Okay, so Parker has a question. She's writing in the chat, and Parker's question is she wants to know why is tapetum multinucleated and has a dense cytoplasm. So before I answer this, any one of you would like to help Parker with this question? Why tapetum has a dense cytoplasm and is multinucleated? Good evening, Iram. I can see Iram has joined in.
Good evening, sir.
Yeah. So any idea why tapetum is multinucleated and has a dense cytoplasm? So this is what Aisha is asking in the chat. Let me, yeah. Can you all see the question on the board?
Yes, sir.
Yep. Okay. Anyone would like to answer? Iram? Yep, go ahead, go ahead.
Yes, sir.
Yeah.
So because in tapetum cells, karyokinesis is not
This is
followed by.
Aisha's question. This is Parker's question. So you're addressing Parker, not me. Okay. Yep. Cool.
Okay.
Cool.
Yeah.
Let's make it. Yeah.
Yes. So in tapetum cells, karyokinesis is not followed by cytokinesis, which makes the cytoplasm really thick. And
So you're saying. So go slow again. What did you say? Karyokinesis is not
followed by cytokinesis.
followed by cytokinesis. So do you understand that, Parker? Means the nucleus divides and cell does not divide. That's why it is multinucleated. But that's the mechanism, right? That's how it becomes multinucleated. What is the functional benefit of a tapetum cell being multinucleated? You're right. If any cell wants to become multinucleated, it has to do the same thing. Okay, it has to divide its nucleus without dividing its without undergoing cytokinesis. Correct, Iram. But why? Why is it multinucleated? And yeah,
because these cells are metabolically active and they store food. What could be the reason?
most of the cells, almost all the cells in a living system are metabolically active, which is a metabolically inactive cell. If any cell is metabolically inactive, it's dead, right? Or it's in cryopreservation or in hibernation, something like that. So living cells are always metabolically active. And you're saying it stores food. So for that matter, many other cells also store food, but they are not multinucleated. If you want to store something, why don't just store in food vacuoles? So why it has to be multinucleated with a dense cytoplasm? For storing food, there are food vacuoles, right? Plants have big vacuoles. You can store food in them. What it has to do with nucleus? So we discussed it in the last class. What does tapetum do? Remember when I was talking here?
Provides nourishment to the pollen grains.
Yes, right. It provides nourishment to the pollen grains. And how does it provide nourishment to the pollen grains? The developing pollen grains? So it is present just adjacent, like it's in contact with those cells, right? Tapetum is the innermost layer, if you remember. And by that logic, it is in direct contact with the pollen mother cell, PMC, that becomes into pollen grains, correct? So first, it is multinucleated because if it is multinucleated, it will have multiple copies of the same, of every gene and chromosomes. It can do rapid and more transcription and translations and can produce more protein, which can be used to provide nourishment to the developing pollen mother cell. Okay. Is it clear?
Yes, sir.
And the dense cytoplasm is because of this very high amount of translation happening in the cells, and lots of protein molecules and other nutritive molecules are present in the cytoplasm itself. Okay. So it's not just storing it for the sake of storage. If you want to store anything for the sake of storage, you don't have to be necessarily a multinucleated cell. You can be a uninucleated cell with a very big vacuole, and you can put everything in the vacuole and store it. But it's storing for use. Okay, use for itself and for the development of pollens, right? It's not for a long-term storage. So does it make sense, Parker?
Yes, sir.
Does that answer your question?
Yes, sir.
Okay, perfect. So it can write it provides nourishment to the PMC, pollen mother cell, which is actually developing pollen. Okay. And since it is multinucleated, there is increased levels of protein translation. That makes the cytoplasm dense. Okay. Cool. That's a good question. So let's go to talking about what we did in the last class. I think so. We started with microsporogenesis. Anyone would like to quickly give me a recap in 5 minutes? Quick. Anyone? Come on, volunteer. Rabia, how about you? Are you there, Rabia? No, yes. Okay, I don't think. Mariam, would you like to give a quick recap? I saw someone raised hand. Yes, Rabia is there. Rabia, would you like to give a quick recap of the last class? Or Mariam? No one? Okay. Aisha? Yeah. I think I'll have to go to Parker then because no one is agreeing for it. Okay, cool. Parker, go ahead. Quickly.
In last class, we studied about microsporogenesis. Microsporogenesis is a process of producing microspores or pollen from a pollen mother cell. Sporogenous cells are diploid, and sporogenous cells undergo meiosis. As a result, it forms microspore tetrads. Each cell in a tetrad is is going to become a pollen.
Yep, correct.
Then we studied about pollen pistil interaction.
Mhm.
In pollen pistil interaction, a chemical interaction takes place between the chemicals produced by pollen.
Right. So chemical, basically, if you're talking about chemical, you can say that the signaling molecule is produced by the pollen and the receptors are present on the pistil's cell surface. Yeah. And then there is this pollen pistil interaction to understand if the pollen belongs to the same species or not. Yeah. Then
In most of the angiosperms, pollen grains are shed.
yes, right.
To cell stage. One is a bigger vegetative cell, and one is a smaller generative cell. Many species shed the pollen at 3 cell stages. The generative cells divide mitotically to form 2 male gametes.
Correct. Correct.
Regardless of the stage at which pollen grains are shed, almost all angiosperms have 2 male nucleus and 1 vegetative nucleus. In a mature pollen, which is ready to form a pollen tube. Then we studied about microsporangium.
A megasporogenesis, you mean, right?
Yeah, megasporangium. Megasporangium can be mono-ovulated or multi-ovulated. Some examples of mono-ovulated are wheat, rice, mango, and multi-ovulated is watermelon, papaya, and so on. A typical angiosperm, Megasporangium's definition is a typical angiosperm ovule is attached to the placenta in the ovary through a stack-like structure called
Right. Yeah. Funicle.
funicle.
And then the junction at which, yeah, go ahead.
The helium represents the junction between ovule and the funicle. Helium in plants is analogous structure to the. It's like
the belly button in humans,
belly
right?
button. Yeah, it's like the umbilical cord in
Yeah.
humans.
Correct.
There is a chelazal end, and sorry.
You're right. There is a chelazal end and there is a micropylar end.
The pollen tubules enter through the micropylar end, and then the chelazal end is like on the other side.
Yeah, that is the base, like the basal plate of the this enzyme. Okay. Okay,
Yes.
cool. So and there are integuments. Integuments are nothing but protective layers, which there are 2 inner integument and the other one is outer integument. And nucellus are the cells, mass of cells that are surrounding the embryo sac. You can see nucellus something similar to as tapetum in the microsporangium. In megasporangium, they are nucellus, the innermost layer, which is in direct contact with the embryo sac. And here, because it's the innermost layer, whenever something is present in contact with something that needs to develop, like embryo sac or the pollen mother cell. The function there is nutrition. So nucellus have abundant food reserves. Okay, they have the food reserves to provide it to the embryo sac for nutrition. Okay, so they're like something similar.
Yes, sir.
Okay, now one thing that you should keep in mind. Yes, yeah,
I have a question.
Yeah.
So the exine, which is the outermost layer of the pollen grain, is formed by the tapetal cell, right?
Formed by the
Tapetal cell, right? Tapetal.
it's not formed by the tapetal cells. The exine is form of sporopollen. It's a biopolymer that is very, it's not formed of cells per se. It is formed of a compound, organic compounds. So a biopolymer sporopollen is the majority of what forms the exine. Okay. So exine is not
But sporopollen
composed
is
of cells.
present.
Yeah, go ahead. Is my voice cracking for everyone?
Yes, sir.
Okay, just give me 1 second. Is it better? Yeah, go ahead with your question.
So sporopollen is formed from the tapetal cell only. Like that's what is written in my school notes. The teacher.
Sporopollen is secreted by tapetal cells, but the tapetal cells, they themselves, because see, tapetal cells are degradable, right? We say that the exine is not degradable. So of course, because it's a biopolymer, it has to be formed from somewhere. It's obvious, right? Do you understand what I'm saying?
Yes.
But the cell that forms it, for example, if you say that tapetum is
Voice is happening.
we also know that there's not an NCRT for pollen. 1 second. Give me 1 second. Let me adjust and see. Is it any better now? Is it any better now, people?
So can you speak?
It's showing full network. I don't know why. Yeah, is it better now?
Yes, sir.
Okay, let me try to shift to the other one. Hello, am I audible now?
Yes, you are.
Okay, is my voice better now? Not cracking?
No, sir.
Okay, perfect. Yes, so I was answering your question, Iram. I was saying that since we know that sporopollen
They are not audible.
Okay, am I audible, people?
Yes, sir.
Okay, is this better? Sorry for this. The network is pretty bad here today. Is my voice any better?
Yes, sir.
Yes.
Okay, so I was saying that tapetal cells can also be of different kinds. This is what is not taught in NCERT. So it's true that tapetal cells, some of the tapetal cells, which are specifically called as secretory or glandular tapetal cells, okay, these secrete sporopollen. And there are other kind of compounds that go into exine, like there is something called pollen kit and other proteins, and also Ubish bodies, which makes exine, which are also part of exine. So all these chemicals are produced by glandular tapetal cells, but it's not like these tapetal cells themselves form the exine. It's not the layer. Cell layer is not exine. You understand? When it's secreted, it gets deposited, and that forms the exine layer. Does it make
So,
sense?
sporopollen is an enzyme.
Sorry, not exine. I'm not saying enzyme. Exine. Sporopollenin is a bioorganic compound, a biopolymer.
So, how is it
So it's not just
transforming to the hardest? How is it transforming to an enzyme?
how it is.
Transforming to enzyme.
How it is transformed into exine.
Yes.
Yeah. So it is. So just like, have you seen starch depositions? Starch or cellulose, it get deposits, right? Or carotenoids. These gets deposits layer after layer after layer, right?
Yes.
So similarly, sporopollenin has this ability to get deposit in layers and make a very rigid structure, because it's a biopolymer. So it will be a long molecule which will have ability to form bonds with other similar kind of molecules and keep getting deposited and making a layer. Do you understand that?
Yes.
Yeah. So so we know that how it should be taught to you is that the tapetal cells, specifically the glandular and secretory tapetal cells, all of the tapetal cells do not do that. So there are category of tapetal cells which are called glandular tapetal cells, and they are the key player in forming sporopollenin wall of the exile. So they secrete and it gets deposited. They secrete and it gets deposited. Okay. Does it make sense?
Yes.
Yeah.
Yes.
So yeah. Cool. So there is some details about it which at this level I'm confused. Should I spend time on it or not? But just remember that tapetum is multinucleated, whereas nucellus, which is asked in entrances, which forms similar kind of functions, like, but this stores, it is a reserve. It is not like it does. It's cytoplasm is not as dense as tapetum. So nucellus is not multinucleated. It is uninucleated and has a ploidy of 2n, unlike tapetum. Okay. I'm aware that some books do write that exine is the exime is formed from tapetal cells condensing and forming a layer. But that's not entirely true. The cells itself are big, but the enzyme layer is very thin. So either you squish the cell and make a layer, or you only take the excretions that secretions from the cell and they gets deposited to make the layer. Much of the evidences say that the outermost layer of pollen is made up of sporopollen, and which is secreted just like any other polymer from a secretory cell, and that happens to be tapetum. Is it clear, and everyone? That's a good question, but at this level you should understand.
Yes, sir.
Cool. Okay, so today let's talk more about Megasporogenesis, just like Microsporogenesis. So what was the key features, if you remember, of the Microsporogenesis? Anything remembered about Microsporogenesis? Quickly.
Producing pollen or pollen mother cell.
Yes. So which cells will give rise to PMC? Something becomes PMC and then, yeah? Tell me.
Sporogenous tissue.
Sporogenous tissue becomes PMC, and then each PMC develops into a pollen. So in a tetrad, remember I told you about the tetrad?
Yes, sir.
What was the tetrad called? Microspore tetrad. So each cell in the microspore tetrad becomes a pollen mother. Each cell in the microspore tetrad becomes a pollen mother cell and then becomes a pollen. But if you remember, in humans also, at one time, how many ovums were produced and released? Were they multiple?
No, only one.
Yeah. Just one, right? Anyone is saying something?
No, sir.
Okay, I thought someone is asking a question. So just one ovum was released, right? And formed and released. Something similar happens in Megasporogenesis as well. So if you define it, first write down, it is the process of formation. Write down. Process of formation of megaspores from the megaspore mother cell. MMC, megaspore mother cell. So here also what happens is there is a formation of megaspore tetrad, just like microspore tetrad. Here also tetrad will form because it's following a similar kind of route. Sporogenous tissues, sporogenous like tissues, here we also call them the same sporogenous tissues. They divide meiotically to form megaspore tetrad. But instead of all the 4 cells of the tetrad becoming a ovule, only one of the cell of the tetrad develops into a ovule. Okay, so you can write down in megasporogenesis, a single megaspore mother cell a single megaspore mother cell usually differentiate into a ovule. Usually differentiate into a ovule. only one of the megaspore is functional. What happens to the rest three?
Degenerates.
They degenerate. Okay. Then you will ask, why were they formed in the first place? Can anyone tell me why? Why evolution? In evolution, this problem cannot be fixed. Why are we wasting? We are making something and then we are wasting it. Can there be any other way to do it? Actually, no. Okay. So why? Because you have to make a haploid cell to make a gamete, right? The female gametophyte or the female gamete, which is the ovule, is also haploid. Now these sporogenous tissues, here the microsporogenous tissues, are all diploid. So there is only one way to make haploid cells from diploid cells, which is meiosis. And the problem with meiosis, or you should say the characteristic of meiosis, is that it gives rise to 4 haploid daughter cells, but we don't need all. So we just, plants don't need, female part of the plant does not need all 4. So it just develops one megaspore into a functional megaspore, while other 3 degenerates. Okay. Write down. Only a single megaspore mother cell develops. And what does it becomes? It develops into, we call it the functional megaspore. And the functional megaspore develops into embryo sac. This is where the pollen tube will penetrate. And if you, all of you have NCRT with you at the moment? I don't think I can pull out that image.
Yes,
yeah, okay. Can you come to this figure 2.8?
So I have a question.
Yes, please. Yeah.
So out of 4 megaspores, how does the plant get to know out of 4 which one is the most capable of giving rise to an ovule?
All are capable biologically, but here the question is of convenience. That's a very good question you asked, actually. So thanks for this question. So Hiram is asking, out of 4, how it will it be random that any one will develop and other 3 will degenerate, or is does it have some selection criteria? So it does have some selection criteria, and the criteria is, that's why I'm asking you to come to figure 2.8. Just open figure 2.8. You will see that the way these megaspore, the megaspore mother cell divides and the tetrad is formed is in a elongated fashion. So you will have multiple cells like here at the corner, and these cells will be the nucellus, right? And inside, like surrounded by it, will be a megaspore mother cell that will give rise to tetrad 1, 2, 3, and 4. Okay, these are the 4 cells of the tetrad. Okay, the outer layer is nucellus. Can you see this in the figure 2.8?
Yes.
And these black inside is the megaspore tetrad. Now, Iram is asking, how do we know which one of the 4 cells will develop into embryo sac and the others will degenerate? So this end is the micropylar end, if you remember. Okay, so the closest cell to the micropylar end, which is the first cell that's closest to the micropylar end, will develop into an embryo sac, and these 3 will degenerate. So it's a signaling based selection, the cell which is closest to the micropylar end, because that makes sense. Then you don't have to displace. You can just degenerate these. You don't have to shift any other cell, because the cell which is developing is already at the micropylar end, so it can start growing. So when these 3 will degenerate, this space will be empty and it can start growing in this space to make the embryo cell. Is that clear?
So, what will happen if four of these megaspores will form into
If four of these
megaspores will form into an ovule?
into
All
an
of the
embryo
four.
sac.
Mm-hmm.
So normally it doesn't happen, but since biology is not foolproof, I'm not very sure if one thing is you will end up having multiple embryo sac. So normally plants go with this mechanism, and hence they are called, this is called as a monosporic development. Okay, and if 2, 3, or all 4 starts developing and there is no degeneration, then it will be a multisporic development. But I'm not sure which plants undergo multisporic development. They can
No,
be
I was just
yeah.
I was
You were just
asking, as an hypothesis.
you were asking because
So, as an hypothesis, I was just asking.
yeah, right. So see, the same question we can ask for any other mechanism where there is a planned death of certain cells because they are not required. But since these are in textbooks, I will not say based on like experiences in research, there are plants that deviate from the normal mechanism, right? So your question is valid. It can happen as a flaw, but if it happens, it will be a multisporic development then. And that's what I'm saying, that I'm not aware of, I don't know the names of any plant who does that. Mostly plants follow monosporic development, where only one of the 4 megaspores in the tetrad develops and the rest degenerates. Okay.
Yes.
That's a good question. Yeah. But let me tell you, it's not that if all 4 will develop, there is a common myth that misunderstanding that if all 4 will develop, there will be multiple ovules. That's not true. Okay. You can still have multiple ovules because there will be multiple megaspore tetrads, and from every tetrad, one will develop, rest will degenerate. Still, you can have multiple ovules. Okay. With degeneration taking place, we can still have multiple ovules. It's not like if all develop, it is multiple ovule, like a papaya, and if only one develops, it is mango. It's not like that. Does that make sense?
Yes. Thank you.
Okay. Is it clear to everyone else? Okay. So the functional megaspore develops into a embryo sac. And this is very important. So the structure of embryo sac is important for you from the perspective of boards and also for entrance. Now, just write down a few points, highlighting Iram's question. The method of embryo sac formation from a single megaspore. The method of embryo sac formation from a single megaspore among the tetrad is known as monosporic development. Okay, monosporic development. Okay. Now, you have to also understand, just give me 1 second. Yeah, am I audible, people? Someone at my door. Am I audible?
Yes, sir.
Yeah. Now, what was I saying? Yes. So let's try to understand how this embryo sac is formed and what is its characteristic, because that's very important for your boards and entrance both. So first thing you have to remember is that angiosperms mostly have this 7-celled and 8-nucleated. Yes, can I repeat what? The line that I dictated, are you talking about that, Parker? Yes. So I was saying that this kind of a process, this method of embryo sac formation from a single megaspore, this method of embryo sac formation from a single megaspore among the tetrad. So there is a tetrad which is 4 cells together, but only one forms the embryo sac. So this is known as monosporic development. Mono means single, sporic. So it's coming from a single cell. Ultimately, the embryo sac in the angiosperms is a 7-celled, 8-nucleated structure. Okay. So I'm deleting this part here so that there's no confusion about the structure. This was just to tell you. Okay. Now, how does one single cell reaches this structure, which is a 7-celled, 8-nucleated structure? So basically, all these 7 cells come from that single megaspore cell from the tetrad that wants to develop, that is selected to be developed into an embryo sac. So first what happens, that functional megaspore, so we start with functional megaspore. And that functional megaspore divides mitotically. Okay, so mitotic division. Now you know that mitotic division is all equal, right? And before we do this, can you tell me what is the ploidy of the functional megaspore? What should be the ploidy of functional megaspore? What was the ploidy of the sporogenous tissue from which it was formed? N was the sporo. N of
No,
what?
I'm asking about functional megaspore.
Yes. So functional megaspore is one of the tetrads. Tetrad is a result of meiosis. And as we know, the result of meiosis is always haploid. So functional megaspores are N, like Iram correctly pointed out. Now, if a haploid cell undergoes mitosis, whatever cell it will form, the result of mitosis is 2 cells from 1 cell. So there'll be 2 cells formed now, right? What will be the ploidy of those 2 cells? So there'll be 2 cells formed, right? Which we known as 2-nucleate embryo sac. So this is the next stage. So from 1 cell, there is a 2 cells, 2 nucleus formed. So the 2-nucleate embryo sac, again, each nucleus will be haploid. Then again, another mitotic division will happen. And remember, just nucleus is dividing here first, okay? And then they go to, so these 2 nucleus go to opposite poles and become 2 nucleated embryo sac. Then 2 more sequential divisions happen. And from 2, it becomes 4. And later, from 4, it becomes 8. So totally there are 8 nucleus that are formed. But the problem is cells have to be only 7. So cell wall formation takes place later. First, just nucleus karyokinesis happens. Do you understand that? Let's take a big. So this is a functional tetrad. Okay. Sorry, functional megaspore. You can also call it the functional tetrad, or the functional cell from the tetrad. So this, let me make nucleus in black. So this is the nucleus, one single haploid nucleus. It divides, and it becomes 2 nucleus. Mitotically, again they will be each haploid. Then again they will divide, and they will become, there will be 4 nucleus. And then finally there will be again a division, and there'll be 1, 2, 3, 4, 5, 6, 7, 8 nucleus. So total 8 nucleus are formed. And then after the 8 nucleate stage, the cell walls are formed. Okay. Is this clear, everyone? So if we go through the stages, you can come to the same figure. I think figure 2.8 only in your book tells about that stage only also. So, but ultimately what happens is these, let me make a big version of it. All these nucleus get arranged. So 3 nucleus go and rest towards the chalazal end. Okay, so let's define first. So this is the chalazal end, and this here is the micropylar end. So 3 nucleus goes to chalazal end, 2 stays in the center, and other 3 goes to the micropylar end. Okay, 1, 2, and 3. Once this arrangement is reached, then comes the cell wall formation. So one wall is formed here, making 3 distinct compartments. So now each one becomes one cell. This is 1, 2, and 3 cells. Here also, there is compartmentalization, 1, 2. These are small cells. And I'll make this one special, the cell which is behind. This is another cell. And apart from it, sorry, apart from it, in the center, we have a big cell where 2 nucleus are together. This is it. Okay. So if you count the number of cells, these 3 are called antipodal cells. There are 3 antipodal cells, all are haploid with haploid nucleus. The cell in the center, which is the biggest one with 2 nucleus, is called the central big cell, central larger cell, or just the central cell. Now, if they ask you the ploidy of central cell, you should not write 2n. The ploidy of central cell is n plus n. So there is a difference in saying 2n, and there's a difference in saying n plus n. Okay, so all 3 are n here. Central cell is n plus n. Do you understand the difference between 2n and n plus n? It's a way of writing. 2n means there is a single nucleus with 2 copies of each chromosome present in the same single nucleus. N plus N means there are 2 different nucleus, each nucleus having a single copy of every chromosome. Does that make sense, everyone?
So can you repeat that part?
Yeah. So if I'm saying 2N, okay, it simply means there is a single nucleus, okay, with 2 copies of every chromosome. Like the nucleus in every cell of my body, except for the gamete cells, is 2N. Okay. So there is a single nucleus in my cell where 2 copies of every chromosome is present. This is clear?
Yes.
But if I say N plus N, it means there are 2 nucleus. That's why I have put the plus sign in between. So there are 2 nucleus, like in this case. Each nucleus will have its nuclear membrane separately, but each nucleus will have only 1 copy of every chromosome. Okay. And if I write just N, again, it means there is a single nucleus. With just 1 copy of every chromosome. Is the difference clear between these 3 notations? People?
Yes.
Aisha?
So this is like very weird because mathematically n plus n equals to 2n.
2N. Exactly. So biology, sometimes we use mathematical notations to explain a phenomena of biology, which does not make similar sense as it will make in mathematics. Okay. So you're right. But you have to understand, if I'm writing N plus N separately, I'm differentiating that there are 2 nucleus, each with their own boundary and just a single copy of genome in it. Okay, 2N and N means single nucleus. Here there are 2 copies and here there is a single copy. Here there are 2 nucleus with single copy. So central cell, whenever they ask you to write ploidy of central cell, the right option will be N plus N. Neither N or neither 2N. Okay, is it clear? Okay. Now these 2 cells at the micropylar end, the most extreme micropylar end, are called synergid cells. Now, synergid cells are very important cells because they play 2 functions. One is when the pollen tube is coming during pollination for fertilization to happen, these synergid cells kind of guide that pollen tube through to the egg cell, which is at the back. Someone has a question? Sir, then why isn't it N comma N and N plus N? Yes, that's right. And actually I have found some books that do write central cell has the notation N, right? But that's not a very standard notation. People have accepted N plus N because we write it plus because they both are in the same cell. And they both are contributing to the protein synthesis or whatever is happening in the cell. So it's additive effect. So biologically, the presence of 2 nucleus, both of them are haploid, but the presence of 2 nucleus in the same cell is having an additive effect on the process of transcription and translation. That's why the sign plus is more standard and justified. But if you write N plus N comma N, that's also not entirely incorrect, but it's just not widely standardized and accepted. Okay. Does that answer your question, Parker?
Yes, sir.
Yeah. Now these synergid cells, first is they guide the pollen. So that's their function towards the pollen tube. And second, they help with nutritional requirements of this very special cell. Why I have made it red? Because this is the cell for which everything is happening. This is called the egg cell. This is in real sense the female gamete. And again, egg cell has to be haploid. Synergid cells are also haploid. So all the cells in the embryo sac are haploid. If you count the total nucleus, 1, 2, 3, 4, 5, sorry, 1, 2, 3, 4, 5, 6, 7, 8. So it is 8 nucleated. But if you count the cell, there will be only 7 cells. So that's why it's called a 7-celled and 8 nucleated structure. Is it clear, everyone?
Yes.
And towards the micropilar end, just in front of the synergid cells, there are some projections which basically, if you ask me biologically where they come from, so these projections are actually thickenings present at the synergid cell only. So this synergid cell has some thickenings at the micropilar end. I'm making the thickening using the same black color. So it looks something like this, okay? Like a projection, finger-like projection. And that is called filiform apparatus. So it actually is a extension of synergid cells only. It is not an independent cell in itself or anything. They are cellular thickenings. And this is the main structure that guides the entry of pollen tube into the synergid. Okay. So filiform apparatus is a cellular thickening of synergid cells towards the micropilar end that guide the entry of pollen tube inside the synergid cell. Is that clear, everyone? Perfect. Now, if you know that this part is micropilar end, I'll just erasing it and writing it a little below here. Micropilar end. And just imagine that this whole structure is present in the this whole structure is present in the ovary, right? So there will be a pollen tube coming like this. So this pollen tube will grow. And remember, I told you that it goes all the way down and enters through this micropilar end, right? So it first comes in contact with the filiform apparatus. And this filiform apparatus kind of helps this tube to enter inside the synergid cell. And if you remember, 3 nucleus are traveling in this tube. Can you tell me what are these 3 nucleus? What is the leading one? It is the vegetative nucleus, right? And these 2 which are trailing are the
Generate.
male nucleus. So now when they are in the tube and they have divided, you can still call them generative nucleus. That will not be an incorrect thing to say, but we very specifically now call them male nucleus because they are male gametes. Okay. Is that clear? So generative and male nucleus. So 3 nucleus travel. So far, so good. Anyone has any doubts?
Yes, sir.
Okay, perfect.
So I have a
So
doubt.
yes. Yeah.
Sorry, I'm frequently asking.
No, it's fine. I always appreciate doubts. It's cool.
So 1 minute. Yes.
Yeah.
Sir, this ovule is completely surrounded by cell wall, right? So how is the three?
How is the?
Sorry. So how is the three the male gametes able to fuse with the synergids and all the three antipodal cells and central cells?
So they never reach till antipodal cells. That's first thing. So they don't have to worry about going all the way through the central cell to the antipodal cell. Okay. And what was your first part of the question? It is they are
So
surrounded by.
yeah, they're surrounded by the cell wall, right? So how are they able to enter the ovule?
So yes. So in the embryo sac, when you go in the embryo sac, these you're talking about the synergid cells, egg cells, right?
Yes, the synergid cell will be able to attract them, right? But you
Yeah.
have a barrier that is the cell wall. How will you be able to cross that?
So the question is, do synergid cells have a cell wall or not, right?
Yes.
Yeah. So the point is, all these cells are haploid, correct?
Yes.
Am I audible, people?
Yes, sir.
Yeah. So the synergid cell wall, the filiform apparatus itself is the cell wall, like that acts. The cell wall is this thickening. What I'm talking about is the filiform apparatus, and this thickening is like a guard, like a bouncer at the gate of any club or any party. So no unauthorized entry is permitted. But how it enters? So it does not have to enter by force only. So pollen tube is, though it's not very very fragile a structure, but when it reaches, the filiform apparatus is actually evolved to help this mechanism. It actually kind of holds on to it because these are finger like projections which are loose and thick, and then they pull it, and also the pollen tube pushes and they pull, and it enters into the synergid cells. Once it enters into the synergid cells, okay, then the egg cell is very easy to access from there, because egg cell does not contain a cell wall. It's a gamete cell, and gametes do not contain walls. They have semi-permeable cell membranes. Does that make sense to you?
Yes, sir. That's what I
Yeah.
was asking.
Yeah, great. So the process is very, very elaborate. There are research papers on it. People are still trying to understand. And some plants have evolved very, very beautiful mechanisms, which people can see in real time. So you can see this pollen tube developing in real time under the microscope, and you can label these with different kind of GFP, green fluorescent, and other proteins. And we are still trying to understand how over the millions of years angiosperms have evolved this beautiful mechanism, which is not present in their precursors. Okay, and even all angiosperms do not show a similar level of what you call complexity. Okay, so and then talking about the central cell is again the largest cell, and that central cell later on will become the, what is one thing which is present inside the seed which is very very important for the development of embryo? When you open a chana, what are you actually eating? What are present in the cotyledons? Endosperm. Remember endosperm, people?
Yes.
And what was the ploidy of endosperm? Do you remember the ploidy of endosperm? Three N, right? Endosperm is 3N structure. Anyways, if you don't remember, please brush it up. But do you see anything here which is more than n? No, right? But there is one cell, which is the central cell, which is n plus n, 2 haploid nucleus. So the two male nucleus, let me tell you, only these two have the ability to fuse because they are male gametes. The vegetative nucleus cannot fuse. So what happens? It's only to lead and then it degenerates. And when it enters into the filiform apparatus, when the tube enters into the filiform apparatus, these two trailing male nucleus are released into the filiform apparatus. They go through the filiform apparatus. One enters into the egg cell and fuses with the egg cell, and n plus n becomes 2n, and egg cells become a zygote. This is clear? Give me a thumbs up if this part is clear. And the other male nucleus crosses the egg cell and goes into the central cell, comes here, then these three together fuse and become a 3n structure known as endosperm. So a 2n and a 3n. And then the antipodals and the synergids, their role is not required later on as it develops into a fruit and a seed. So they die and degenerate. And what you only see inside a seed is an embryo and an endosperm. Does that make sense? Can you now connect the whole picture together? People? Yeah. And this process is called double fertilization. Why it is called double fertilization? Because there are two fertilization events happening. One male nucleus is fertilizing with the egg cell to give rise to a diploid zygote, which will later on become a diploid embryo. And another male nucleus is fusing with the two polar nuclei. Okay, I forgot to tell you, in the central cell, these two nuclei are called polar nuclei because there are two so nuclei, plural. So the other male nucleus fuses with the two polar nuclei, giving rise to a 3n nucleus together, and that forms the endosperm. Is the whole process clear to everyone?
Yes, sir.
So you can write down the synergids and the filiform apparatus, which is an extension of synergids only. The synergids and the filiform apparatus play a role in guiding the pollen tube into the synergid cell. Play a role. And how do they guide? First, they guide through chemical cues, chemical signal. So the pollen tube constantly keep getting chemicals signals from these synergid cells, and that's how it knows where to start bending. Otherwise, it will keep going down, right? So it has to bend and reach the right place. And then write down only the two male nucleus, which are also you can call as generative nucleus. Only the two male nucleus fuse. One fuses with the egg cell to form embryo. One fuses with the egg cell to form embryo. And the other fuses with the two polar nuclei in the central cell. And the other fuses with the two polar nuclei in the central cell to form endosperm later on. To form endosperm later on. Make sense? This process is known as double fertilization. This process is known as double fertilization. Okay, because there are two fertilization events taking place. Two fusion events are taking place. So in angiosperm sexual reproduction, double fertilization happens. And in this double fertilization, the event which is specifically giving rise to endosperm, the fusion of two polar nuclei with a male nuclei. So you can understand that there is three nucleus fusing together. Yes or no?
Yes.
Polar nuclei. So that particular event in double fertilization is called triple fusion. So write down the fusion of three nuclei. The fusion of three nuclei. In the bracket, you can write two polar nuclei and one male nuclei. Two polar nuclei and one male nuclei. The fusion of three nuclei during double fertilization. During double fertilization, to form endosperm, is called triple fusion event. It's called triple fusion event. So this question is very, very common, that explain double fertilization and triple fusion in detail. Or what is double fertilization and triple fusion event? So no one will be confused from now onwards. Everyone is clear? Okay, perfect. Anyone has any questions? Because I'm going to give you a homework then. So there is one easy part in this chapter, which if you people can take care of, we can expedite the chapter to the more important, like we can go into detail of double fertilization and triple fusion and embryogenesis, which is pollination. So your homework is to read about pollination.
So your voice is cracking.
Okay, so there are two types of. Is it any better now, people?
Yes, sir.
Okay. So I'm giving you a homework to read and make notes. On the topic pollination. It is a simple topic, just theory. Sorry. Just theory, where you will talk about what is pollination, types of pollination, so self-pollination, cross-pollination, and the agents of pollination, what causes pollination. So all theory. So will you do this? Will you finish this part yourself by making notes? Everyone?
Yes, sir.
Parker, Iram, I want to see thumbs up from all of you so that no one can then backtrack on it. Okay, cool. So let's end here today. And please come up with your questions on pollination and this thing. We will go into detail of double fertilization and you about hybridizations, okay? Outbreeding, hybridizations, and seed and other things. Okay, so I think I will be able to finish this chapter in 2 more classes if you finish pollination on your own. And then we can go to evolution. Okay, cool. So I will see you all in the next class. Take care. Bye-bye. And we have a WhatsApp. Before you all leave, we have a WhatsApp group. Every one of you is added to that group with me.
Yes, sir.
Okay, so I will be asking all of you, just put your next exam date that's going to happen in your school and the syllabus that's coming. Okay, if you do not know the exact date, just the tentative date and the syllabus that, because I will have to course correct if it needs. I have to check everyone's syllabus and see if some chapter is completely out of the way, and we will find some time to do that. Okay, so everyone will post with their name, their date of exam, next exam, and the chapters. Okay, on the WhatsApp group, and I will share. Parker says they didn't give the syllabus. So like anything, any speculated syllabus you have in mind, or if you do not have the syllabus, just tell me what have been covered in your school. So clearly, whatever have been covered, the